Well, hi, good morning, everyone. I think most of you know me, and I know a lot of you, but it's good to see everybody. And for those of you who haven't met me or haven't worked with me, I'm Quentin Reed. I am the statistician for the Southeast area. I, on paper, I'm part of the area office, but my body resides in Raleigh, North Carolina, where at the ARS location at North Carolina State University. But this is my first time since 2024 getting out to another location, so it's really great to be out here and get to see some of the other science that's being done in the area. And I'll be here for a couple days consulting with people on different projects, and I felt like it was also a good opportunity to do a couple of presentations like this. So yeah, this is one that I was asked to prepare these materials a few months ago for one of the research units in New Orleans. But I think it's valuable for all scientists in ARS. And some of this we're going to talk about experimental design, just kind of a conceptual overview. It's not highly technical, but it's more conceptual. So hopefully it will spark some discussion, and if people have questions, feel free to interrupt. And yeah, just giving a big picture overview. Some of it may be review, and some of it you may learn something. So yeah, I'll have to share screen as well for the folks on Teams. Let me do that. There we go. Okay, so yes, I call this experimental design matters. Like the matter that we're discussing is experimental design, but it also matters. How you design your experiments influences what scientific questions you can answer. So that's sort of the double meaning there. So just to give a quick overview of what we're talking about today, the main point I want to convey, or one of them, is that a scientific experiment is like a model of reality. It's in the lab or whatever under somehow controlled conditions that are not a perfect mimic of reality, but we're trying to model reality. And through this artificial world of the model that we've constructed in our experiment, we're trying to understand some processes, some cause and effect that occur out there in the real world. And as I just mentioned, you have to consider specifically what hypothesis you're testing or what scientific question you're trying to answer when you set up an experiment. And there are trade-offs. A design that is good for answering one question isn't ideal for another. We'll talk about trade-offs, pros and cons, as we go today. And if you put careful thought into how you design an experiment and how you set it up, that can help you extract the most useful signal, the pattern you're trying to investigate, out of the data with a minimum of unwanted noise, and that will help you get the answer to the basic question or the applied question that you care about. So I'll give a little historical and conceptual background on what experiments are for you first here. So an experiment, we can define it as a controlled procedure to compare or test a hypothesis. And we'll talk about control in a little bit. So a hypothesis, in other words, a statement that we can verify or falsify about the world. We want to get some evidence either in favor or against that hypothesis, or we might have more than one idea about how things might be, and we can compare them by means of an experiment. And experiments are especially useful for learning about causality. If we change one variable X, what happens to variable Y as a result of X? Like X somehow mechanistically affects Y. And this is fundamental to how humans learn about the world. Babies learn about the world by experimenting with things. And, if I push this teddy bear, it moves and that kind of thing. So that it is very natural for us human beings to experiment. We've all done it, instinctively pretty much. And obviously the scientific method is more complex than this, but in sort of the very tidy and oversimplified picture of the scientific method, experiments play an important role. The scientific method starts with an observation of nature, and you see a pattern. Well, what process is behind that pattern? You can use the observations you have plus any prior knowledge about how the world works to formulate a hypothesis about what mechanistic or causal process is underlying the pattern, and then you set up an experiment to test whether or not you can get evidence for or against that. I mentioned already, an experiment is a model, a conceptual model of reality. And just like a computer model or a simulation is a model, everything that we do, or a map is a model of a piece of land. It's a simplified and abstracted mimic of something that's in the real world. And so I see experiments as falling along a trade-off when it comes to models of reality, between control of the variables and realism. So like the ultimate control would be like a pure in silico computer simulation. You can exactly set variables in that simulation and parameters to exactly what you want them to be, but it is farther from the realism end of it, right? Because it's only a computer simulation. It's completely dependent on the assumptions that have been used to put that model together. But a controlled experiment where you set all conditions constant except for whatever you're trying to manipulate is kind of, you have pretty good control if the experiment is set up right, but you're not you don't have the realism of going out in the field and making observations. That's maximal realism. You're collecting real world data, but you've sacrificed control at that point. You cannot you haven't randomly or assigned variables, you're just observing them. So you have correlational data in that situation. So, and then there's sort of, some people call natural experiments, where you can kind of, even though you didn't, as the experimenter, randomly assign treatments to experimental units, you can treat it as if it was random. Kind of like Brian's situation where some aquaculture tanks, got a disease outbreak, but it's possible to, assume that it was just effectively, even though they didn't inoculate the tanks with disease, you could basically assume that it was more or less random which tanks got the disease and which ones did not. And you can almost treat that as if it were truly randomly assigned treatment to tank. But of course, you have to be careful. You're on thin ice there if there's some underlying confounding reason why some tanks got the disease and some didn't. So that's an example of a natural experiment. It's somewhere in between a truly randomized experiment and observational data. Yeah, and as experiments are models, this is like one of my favorite quotes of all time. The statistician George Box said that all models are wrong, but some are useful, right? So models are always, in some way, have lost some of the characteristics of reality. Laboratory conditions do not exist in reality, but so it's not a perfect mimic of reality, but it can be useful in the sense that if we can assume that the processes going on in the lab have some relationship to what's going on in reality, it's useful because the experiment teaches us something about reality. But if we can't make that assumption, then we haven't learned a whole lot from the experiment. And as I said, there is ultimately a trade-off between realism and control, and you're kind of, when you're doing an experiment, you're more on the control side, and you've given up some of the realism. So I don't know if you all find this stuff interesting, but I find the history of science to be interesting. So I'll just give a couple minutes of background on, experiments. Obviously, as I said, humans and even animals use experiments to learn about the world. So experiments have been going on since the beginning of time, but formalized experiments as we know them today, we can trace their history back about 1,000 years to an Arabic sort of, the term scientist didn't exist at the time, but polymath or genius in many fields named Ibn al-Haytham, who at the time it was thought that vision worked by light or somehow rays going out of your eyes and striking objects. But he had the hypothesis that really it's somehow something is going into your eyes to make you see. And through some clever design using a chamber with a pinhole in it, was able to test and gather evidence in favor of the hypothesis that light goes into your eyes. And that is the first documented manipulative experiment that we know of, that we sort of trace the lineage to the experiments of today. And then, another landmark in the history of experimentation was in the 1600s in Italy, in Tuscany. Galileo, the physicist, used experiments that he set up, rolling steel balls down tracks of different slopes to test his hypotheses about how forces act on physical objects to sort of upend the paradigm that had been around since the ancient Greeks. A very important type of experiment that we have today is the randomized clinical trial, where some subjects are assigned to receive different medical interventions. And the first one that we know of was in 1747. At the time, the British Navy was plagued by scurvy. After sailors had been on the ship for a long time, they started to get these terrible symptoms, and there were all sorts of folk theories for why that might be the case. And it was obviously of great military and economic interest to figure out how to cure scurvy. And so this was sort of the first known attempt to randomly assign individual sailors to different treatments. So they had, it wasn't a highly replicated trial. They had 12 individuals and 6 treatments. So 2 people each got a treatment. And there were various things that had been proposed, like making people drink cider, somehow making them drink sulfuric acid. Not sure what that was supposed to do. Seawater, a couple other things. One of the treatments, which was sort of anecdotally known to be beneficial, was citrus fruit. And as we now know, scurvy is a deficiency of vitamin C. So, at least in the simplistic story we get, those guys got better and everyone else did not. But, it was not really the sort of flip side to the story that isn't often told, is that it was not understood why the oranges and lemons, because vitamin C had not been isolated yet, so no one understood why that was making them better. So they, started to do things like, they thought it had something to do with the purity of the lime juice, so they would boil the lime juice so that it could preserve longer on voyages, but that destroys the vitamin C. So it took more later experimentation and studies to truly understand why this was the case. But it did establish the association between citrus and scurvy. And that was sort of an ad hoc experiment. Like I said, it didn't have a lot of replication, and it took a while that didn't really kick off a revolution in experimental design. It took another at least 100 years until experiments became more formalized and systematic. It was really in the late 19th century that the principles of experimental design, as we now follow, were set down. There was an important milestone there was Charles Peirce, who set up randomized controlled trials in psychology, basically testing how sensitive people's fingertips were to pressure, and it was set up in a randomized design with repeated measures within individuals and across individuals. So, this design would sort of pass peer review today, whereas previous ones might not. And obviously, as ag scientists, most of you must be aware of R.A. Fisher's pioneering work. He sort of single-handedly not only invented I wouldn't quite say invented, but put all together in one place the principles of frequentist statistics that are still used today. And he was working in ag science, in agronomic trials, like a lot of you work on, and that's sort of why the lingo of experimental design sounds agricultural, like plots and blocks. The people that are not actually setting up trials in fields still use that terminology, and that's because Fisher developed it within the field of agriculture. And continuing from there, as I said, the scurvy trial in the 1700s was kind of a one-off. It took until probably the 1940s until randomized controlled trials were set up in a formalized and modern way. And a tuberculosis antibiotic trial was kind of the first one that is considered to follow those principles. And before that, most medical journals consisted almost exclusively of anecdotal studies, right? Like case reports where a doctor would have an interesting patient and diagnose them, come up with a cure, see if it works, and write it up. And that can still be incredibly useful information today, and there still are those kind of case reports are still regularly published, but that was all there was until the 40s. And obviously that is not really adequate to show the efficacy of medical interventions in a lot of ways. So obviously it's very important to have those randomized trials. And experiments also started to be used in industry, and that was really pioneered by Japanese firms, starting in the post-World War II era, using experiments and then subjecting the results to statistical analysis to do quality control on products. And then, it's at 2025, now it's 2026, but we are now sort of, obviously, everyone's talking about big data, AI, and so forth, and experiments, because of all the infrastructure you have to set up and the conditions you have to control, is not necessarily producing big data. We have relatively small sample size and amounts of replication compared to the high throughput observational data you could collect in a non-experimental setting. So is the paradigm of experiments still useful within that world, right? I think it is, and it can be integrated with these other approaches to learn a lot. Yeah. So that was a little whirlwind tour of the history of experiments. Does anyone have any questions or anything before I proceed? Right. I see your minds are also blown by how interesting that was, that you have nothing to say. Okay. Now, the rest of the talk is more on the technical side of how experiments are set up and looking at some of the principles behind that. And there's kind of a bit of a glossary. We're going to be talking about a lot of these. By defining the terms one by one, it will help you understand how they're all related and how experiments should be designed. So let's start with the term treatment. What's a treatment? Can anyone throw out a definition of treatment? Okay, yeah. So, and not only is it a specific condition, it is a condition that we manipulate or impose on individuals in an experiment. It's called treatment, is kind of a term from the medical realm. You're treating a patient with a drug, but we call everything treatment regardless of field. And many treatments in experiments are in the form of factors. Different treatments are different levels of a factor. A factor is a variable that we do not necessarily measure. We actually set, the experimenter sets the levels of that variable. And, you know, obviously, many different things could be factors, like if medication is the factor, the different treatments would be different levels of dosage, or, you know, management regime in an agronomic study could have different levels, different types of management. Okay, and here's another quiz, pop quiz. Can anybody define what is a control in the context of an experiment?
Untreated variable.
An untreated variable. So yeah, essentially it is a control is an observation that you take as part of an experiment that shows the effect of no treatment, or, you know, sometimes you have to do a mock or placebo treatment. And that establishes a baseline to which we can compare to look at the difference between the control and treatment. We interpret that as the effect of the treatment. Now, what constitutes a control? There's no one answer to that, because it depends what you're controlling for. And in an experiment, you're manipulating one or more variables. Everything that you manipulate in experiment ideally should have a counterpart that is not manipulated in a control group. And there may be more than one control. In particular, I'm sure most of you are familiar with the concept of positive and negative controls. A negative control, like I said, is a baseline, non-manipulated group that we can compare the treatment effect to. But there are also positive controls where we do expect an effect, and that ensures that the procedure is giving expected results using standard methods. Like we could, if we're trying to test a new vaccine, you know, the control group would not receive any vaccine. Then you give some individuals the experimental vaccine, and then a positive control would be a known effective vaccine that we know should provide immunity. We can then see how that would serve as a positive control. So we can see how well the new one is doing. And it may not always be possible logistically to have a separate unmanipulated control group, and in some cases ethically as well. For example, like in mouse studies, when you study methods to heal, you know, broken spines or dislocated vertebrae, it's essentially known that if you break a mouse's spine and do nothing, that they will not get better spontaneously. So typically those experiments do not have unmanipulated controls, like for both logistical and ethical reasons. But in some cases, you can use pre-treatment measures on the same individuals that later get the manipulation to serve as controls. Okay, experimental unit. I won't make you all define every, I was gonna ask you to define every term, but I'll just tell you what an experimental unit is. If you don't know, it is any individual or thing or object that we independently, randomly assign a treatment to. You know, like if you're testing different plant genotypes, the experimental unit would be the plot of land, the area where we randomly pick one of the genotypes that we're trying to compare between to plant on that piece of land. So the piece of land itself, the plot, is the experimental unit. Or, you know, if we're doing, vaccination study or some other medical intervention, it would be the individual organism that we randomly assign, are you going to get the vaccine or not? And, the experimental units can be very large. Our colleagues in the Forest Service often do studies on the scale of an entire watershed. The entire drainage basin is randomly assigned to have a certain logging, be clear cut or selectively logged or what have you. So those they can be huge, or it could be a tiny flask, it just depends on the study. Now, an important distinction is that the experimental unit is not always the same as the observational unit that we make measurements on. Like if you had, if your experimental unit is the tank, you have a bunch of fish in the tank, you're measuring the weight of each fish. Those are the observational units. But each fish was not independently randomly assigned to a treatment. The whole tank got a treatment. And that's where there isn't always a one-to-one correspondence between experimental unit and observational unit. Or you could have repeated measures in time on the same experimental unit. So you could have multiple observations per experimental unit. And we use various statistical models to account for those discrepancies, mixed models. That's where that comes into play. I'll talk about that a bit later. So how can we break down experimental design into different components and then think about how each one should be set up? There's sort of three components to a structure of an experiment. And this is some stuff you'll see in the literature. And I'll go through each one. One is treatment structure. What are the treatments? How are they applied and how they relate to each other? Design structure is how the treatments are assigned to experimental units and what is the relationship between the different experimental units in space and time. That's where blocking and stuff comes in. We'll talk about that in a minute. And response structure, what responses are you going to measure and how in relation to the units. Like I said, you could measure, if plot is your unit, you could measure something on multiple plants in the plot or multiple fish in the tank, repeated measures and that kind of thing. And, all this stuff about design also applies to observational studies, even though the treatments are not randomly assigned. The same ideas apply of like relationships in space and time and how you measure things and so forth. So, the statistics end up being similar. It's just the conclusions that can be drawn differ, whether it's an experimental manipulation or not. So let's talk about treatment structure. This is the different terms you might have heard before, like a one-way design or factorial, regression design, response surface. Those are all different treatment structures. A common one that we see a lot in ARS is a factorial design. That's where you have more than one factor that you're manipulating, that each one can have different levels, and usually the experiment includes combination of levels across factors. You could have a fertilization treatment with multiple levels and an irrigation treatment with multiple levels, and then cross that. So different plots each get a combination of the two factors. And G by E interaction, obviously that's a very common, factorial is set up to look at interactions between the factors. G by E is a real common one. A common garden experiment is a typical type of factorial design where you have a bunch of different environments. So those are different levels of environment. And within each environment, you have the same set of genotypes that you measure traits on. And that, because we have all combinations of genotype and environment, we can estimate genotype by environment interaction. And obviously, in the real world, things are subject to all variables, all influences from the environment at once. It's not just one variable. So you have the more interactions you want to include in an experiment, we could argue that's more realistic because, many different things can be changing at once in the environment. But, once you get to very high order interactions, 3, 4, well, first of all, it's the curse of dimensionality. You have to multiply the number of combinations. So the number of samples you need total blows up. Not to mention, it's sort of hard to interpret very high order interactions, what they mean exactly. So again, it's a trade-off, right? Just a one-way design is very simple to interpret, but it doesn't have as much realism because that's only one of the many variables that actually could be changing in the real world. Okay, so dating back to R.A. Fisher, there are basic principles that should be taken into account when designing experiments. The first one is randomization. This is critically important. Which units, which individuals, plots, whatever, receive which treatments should be random. If it's not, then that can cause an overestimate or even an underestimate of the treatment effect. So there should be any unit that's entered into the experiment should have an equal chance of getting any of the treatments. And that seems like we take it for granted now, but in the 1920s, it was sort of almost like controversial that Fisher used a deck of cards and shuffled them, like pulled cards from the deck to decide which treatment different plots of land would get. And I was like, well, that's not very scientific to have a deck of cards out, but yeah, now we see that as a bedrock of experimental design. Replication being the second principle, we want to estimate variation between treatments. That's the signal we're trying to pull out. But we also need to look at the variation within treatments, the noise essentially. And so to evaluate our signal against, and in order to look at variation within treatments, you need multiple experimental units, replication within treatments to estimate that. And the last one, not as commonly thought of as randomization and replication, is local control. That means conditions should be as constant as possible across the treatment groups, essentially that we're holding other variables besides the one we're manipulating as constant as we can. And, that can be done by having very good protocols and, doing everything cleanly so that you minimize the variation, but there's always going to be some that you can't eliminate. So we do our best through statistical models to account for any variation that we can't, physically eliminate. Yes, Ryan. Yeah, go.
I deal a lot. I never heard the term local control before, but I like it because I do it all the time.
Right.
And it seems like that's always at odds with randomization. Seemingly, it feels like it. And is there like, is that just like a qualitative assessment how you push one thing into more local control versus randomization, or is there any?
So what do you mean it's at odds with randomization?
Well, it's like if you use like this room here, for example, and there are a bunch of tanks instead of tables. The tanks on the inside of this row here always perform worse because there's people walking up and down all the time. And so if you randomize that and end up with a bunch of one treatment in the middle row, they're always going to perform worse.
That's right.
And so I want to like local control against them.
Yes, and so that is where blocking comes into play. You wouldn't want so if there's some gradient of vibrations in the floor that's bad for the fish, you don't want to put all of one treatment at one end of that because it's going to cause confounding variation. And so the sort of most common way to deal with that is to use blocks to structure, to constrain which units can receive which treatments so that it's evenly balanced across your environment. And that increase, we will talk about that shortly. So yeah, thank you for the question. And when we're talking about experimental design, a term you'll see in the literature is optimal design. And it's called optimal because if you have some criterion from a statistical model, like a test statistic you're trying to estimate to test some hypothesis, given a particular sample size that you have, there's going to be a design that is optimal, that has maximum statistical power to pull out that signal. Yeah, so given the resources you have and given the constraints you have, we can always find a design that gives us maximal statistical power, to answer that question. Not absolutely, it's not necessarily absolutely high power, but it's the highest we can get with the resources we have. And in the stat literature, you also see the term efficient that kind of has the same meaning. And so the optimal design is one, or the most efficient design is one that maximizes the ratio of signal variation that we care about and are interested in to noise variation that we're just trying to account for that we're not necessarily interested in. And the two things that you want when you're estimating something is you want estimates without bias. You want to be as close as possible to the truth, and you want minimal variance. You want to have low uncertainty in your estimates. And we'll talk about how whether a particular design is optimal or not depends on the specific hypothesis you're trying to test. There's no one size fits all design that's best in all cases. Otherwise, I wouldn't have to be telling you all these nuances. Okay, so let's actually talk about some of the different experimental designs that are out there and compare and contrast them. So yeah, this maybe, Brian, will help your thinking on this stuff. The sort of simplest one is the completely randomized design. And the CRD is called in the literature. There's no grouping or any structure whatsoever of experimental units. Every one is independently randomized. Like if you had, for these slides, I have an example where you have 3 genotypes and 2 treatments. It's a 3x2 factorial design. So there's 6 combinations of genotype and treatment. And we're going to want 4 replicates of each of those 6 combinations. So 24 units in total. And let's say they're laid out like they're plots of land that are spatially laid out in a grid 6x4. CRD would mean, for any one of those 24, there's a perfectly equal chance of whether it would be assigned to any of those 6 combinations with no structure. So yeah, you could have a randomization where all of the ones from one combination are like over on the left or whatever. That's certainly possible. And right, you don't have, this is great for randomization replication, but you don't have very good local control if you have a completely randomized design, because the error term in your statistical model cannot distinguish between variation due to treatment and variation due to environment. They're bundled together in the error term. Now, this is fine if environmental variation or spatial variation is very low. Like if this is like a growth chamber or a greenhouse or something that has a very constant environment from one side to the other, then it really doesn't, the local control really doesn't matter that much, and a completely randomized design is fine because there's no blocking, so you have higher degrees of freedom, and that's a good thing if we can not worry that much about heterogeneity. But it is not the most efficient design for more natural settings like a field experiment. There is, now if you do have a CRD and you do have the spatial locations of the units, you can kind of work around the fact that there's no blocking by including a spatial random effect in your statistical model, which will enable you to pull apart the environmental variation from the treatment variation. So that's a workaround. But if your design is not spatial, like if they're blocked in some other, or if they're organized in time or some other way, you can't necessarily do that. So how do blocks help us fix this issue? Well, it is a block. It's a group of experimental units that are somehow related to each other. They're closer in space or they're closer in time. Somehow we think that they are more similar to each other than they are to the other experimental units in our experiment. Right, like I said, they could be some plots that are in a group and are right next to each other, for example. And why do we use blocks? They can be used to account for other environmental variation or influences besides the thing that we are actually trying to test experimentally. And this can actually give us more statistical power, a higher chance to detect a true effect that exists than we would have if we just did a straight up complete randomized design. And why is that? Or well, first, yeah, just conceptually, the examples of blocks would be like if you divide your field up into regions and have multiple plots in each region, each of those little groups is a block because they're right next to each other, their environment is more similar than it is to ones elsewhere in the field. You know, maybe the drainage is different in that part of the field, or the soil has a gradient of, you know, chemical composition or whatever. Or, you know, in human studies, you might want to do some educational intervention, and you say, okay, I'm going to do this study at multiple schools. Classrooms within a school are related because they share the same influences of being in the same school, but we're going to like give one classroom a control group and one some educational intervention within each school. So that's a block. The school is a block. Or hospitals also would be an example where you have many patients at each hospital, some of which get a drug and some get a placebo, but the hospital is a block because patients in the same hospital, you would think their responses are more likely than not to be similar than patients from different hospitals due to, we don't necessarily have to say why that is, but we just think that that is the case. So the most common type of block design that certainly that we see in ARS is a randomized complete block design. That's called RCBD. So this is the same. Let's say we had our 3x2 study with 4 reps, just like in the CRD example. Now you can see there's 4 sectors that we've divided this area into, each of which is a complete replication. It contains every one of the 6 treatment combinations exactly one time. Yeah, so they didn't have any other text there, but that's the randomization is constrained. We randomly within each block, it is random which one of those 6 plots got each of the treatments, but we've constrained the randomization so that it's exactly balanced and there's exactly one of each treatment combination in each rep. And a split plot is a kind of a subset of a randomized complete block design that you see a lot in ag studies. Split plot will be done if there are 2 or more levels of randomization. So if there's 2 or more factors that you're testing, you can do a split plot where one of the treatments is randomized at a larger spatial level. That's called the main plot, and then another at the subplot level. Like in this example, each block is divided in half because we have 2 treatments. One half is randomly assigned to treatment 1. The other half is randomly assigned to treatment 2. And within each of those halves, we divide that up into subplots, which are randomly assigned to the different genotypes. It's still a type of complete block design, right? Because each block still has exactly one of each of the combinations of the 2x3 in this case. And sometimes people use split plots for logistical reasons. If it is logistically difficult to, like if the treatment is some kind of irrigation and you can't have like a separate irrigation at that small of a scale, you would have irrigation could be the main plot factor, but it's easy to plot plant genotypes in very small plots of land separately. So that would be oftentimes your subplot factor. Now, it's not only good for logistics, it can actually help your statistical power in some cases. The split plot has more power than a pure randomized complete block to detect the effect of the subplot factor, the one that is randomly assigned at a smaller scale, and the interaction between main plot and subplot. It has more statistical power than an RCBD, but it has less statistical power to detect the effect of the main plot treatment effect. And that makes sense to me because you've assigned let's say the irrigation treatment to less independent random areas, right? There's only eight main plot or eight main plots within each block, within all the blocks put together, but there's 24 subplots there. So there's more units that got randomly assigned to genotypes. So there's more power to detect a genotype effect compared to the treatment or whatever it is. Make sense to everyone? So yeah, it's both logistical and it can be scientifically or statistically preferable. I like to say that there's trouble in randomized complete block paradise because honestly, like, I feel like ARS scientists are in love with RCBDs. Like, whenever they set up an experiment, it's like, okay, the first thing that goes to their mind is RCBD, and they love that design. But it's not always the best, and there's other ones out there that are kind of underutilized by ARS scientists, and not just ARS. I shouldn't single out ARS, but scientists in general. Because why would you care? Well, because you can actually get a more efficient design with more statistical power for the same amount of effort, the same amount of replication and experimental units if you might set it up in a different way. So why it's sort of a win-win situation. What are some of the other designs besides RCBDs that may be preferable? An incomplete block design can be better than an RCBD. That's a block design where each block is not necessarily a complete replication. It's only got a subset of the treatments in each block. Like, there's no rule or law that all treatments have to be in every block. Especially because, like, it doesn't make sense to me that, blocks are supposed to be, within a block is supposed to be environmentally homogeneous, and among blocks is where you should have the variation. But the size of an area that exactly fits one complete replication, one plot in every treatment, why does that have to be the same size as a maximally large area that has negligible environmental variation within it? It doesn't. There's no reason why those things should correspond in any way. Like if you have a 2x2 study, there's 4 plots per block. But then what if you had a 2x4 study? There's 8 plots per block. They're twice as big, so they should be more environmental variation within that block than if it was just a 2x2, right? So, and especially if you're doing some like trial where you're comparing hundreds of genotypes, for example, then if you have a complete block, it's going to end up being huge, and it might be like half your field. So is that really a safe assumption that there's no environmental variation within that? I would say not necessarily. And like I said, these if you set up the incomplete block design properly, it can give you higher statistical power than an equivalent complete block design with the exact same number of replicates. So the sort of ideal incomplete block design is a balanced incomplete block design. And why is it called balanced? Because every treatment should appear equally as often within the same block with any other treatment. That's what balance means. And you can only get perfect balance with like certain integer combinations. Like if you have sort of an odd number of treatments and blocks, you're not always able to get a perfectly balanced design, but there are algorithms that can get you as close as possible to perfectly balanced. So this is an example of one where, like, okay, we have 7 treatments and 7 blocks with 3 replications of each treatment. And you can see that the way it's set up, each treatment appears in a block with any other treatment exactly once. Like in treatment 1 is in block 4, it's with 4 and 7. In block 6, it's with 3 and 6. And in block 7, it's with 2 and 5. So it appears exactly once with all the other treatments. And if you look, that's the case for all the other ones too. And yeah, so then that's so 3 and 7 happens to be a combination that you can set up a perfectly balanced incomplete block design with. And yeah, so because we have more blocks, there's only 3 reps here, right? So if it was an RCBD, it'd have only 3 blocks, but now we have 7. We can more finely pick out environmental variation with 7 blocks. That's right. Yeah. So there it's yeah, I think I didn't randomize, it's just an increasing order every time, but you would within the block randomize the order as well. There's also disconnected incomplete block design, which sometimes may be necessary logistically, but it's not, it's inferior to a balanced incomplete block design. Here, each block gets a set of treatments. You can see here that some of the blocks have treatments 1, 2, 3, and some of the blocks have treatments 4, 5, 6, but like treatment 1 and 4 are never together in a block, for example. And usually this is not as good as a balanced design because it's harder to like estimate the difference between treatment, for example, treatment 1 and treatment 4, because they never appear in the same block together. You still can, if we treat blocks as random effect, you still can, but if you it's better to have a balanced design. This is one that you should typically avoid unless it's logistically necessary to do it this way. Yeah, this is sort of a bit of a whirlwind tour, but another one that you see a lot is the augmented design. And in an augmented design, some of the treatments are replicated, and we call those checks. And you can see here that the orange ones, check 1 and check 2, follow an RCBD design. In every one of the 6 blocks, check 1 and check 2 is present. So that's like an RCBD with a randomized complete block design with respect to the 2 checks. But most of the other treatments, like maybe the genotypes we're testing, are unreplicated or they have fewer replicates than the checks. In this fake example, there's only 1 of each of the non-check treatments in the entire experiment. And here, but we can still figure out the block effect because we have some of the same checks, which are often like standard varieties or genotypes that we know their properties very well, that the error from those are used to evaluate the effect of the treatments or genotypes that are not replicated. And this is useful, for example, in breeding trials where you're evaluating dozens, hundreds of genotypes. It isn't logistically feasible to replicate every single one of them. So often this design is used in those contexts. And there's other ones. I won't get into I'm sort of running behind a bit, so I won't get into the details of some of these other designs, but just to make you aware of them. Some, like I said, some we use because they're actually statistically better. They're more efficient, and some because of logistical or physical constraints, we have no choice. So like a strip plot design where you have 2 treatments applied crosswise to each other. Treatment A is applied to the different columns, and treatment B, the different levels on the different rows. A row column design has blocking in the row and column direction. A special example of that is a Latin square. If you like to play Sudoku, you'll find this one good because a Sudoku board is also a Latin square where all the rows and columns have 1 of each of the treatments. And yeah, that's actually very efficient given the amount of replicates it has, and is also underused, I think. Alpha lattice, you may have heard that term, is a kind of incomplete block design where the blocks are incomplete, but they are grouped so that each group of blocks has 1 complete replication. But it's sort of set up so that it's balanced, where each treatment or genotype appears together in a block with with each other one as evenly as possible. And the blocking is something that I've sort of been talking about plots in a field that's blocking in space, but it doesn't always have to be in space. There's also blocking in time. It's not just for spatial arrangement. Like if you have a growth you only have one or two growth chambers, but you run the experiment multiple times, like in batches, those are considered blocks as well. They can be, or like in livestock trials, they often use a Latin square design where the same, because you don't have that many, it's expensive to have a lot of replicates of cows. So the cows are given different diet treatments or different interventions in a different order in time in each one. And that's often follows a Latin square design. So it's very efficient with few subjects. All the stuff I've talked about, the experimental designs can be repeated in space or time. Like if you have multiple environment trials, possibly over multiple years, you're testing the same set of genotypes. Maybe within each environment, there's blocks, but then the whole thing is replicated in multiple environments, which are either locations or over time or both. And usually, if you have an experiment like that, you would want to do a new or separate randomization. That they still run trains through here, huh? Cool. Oh, awesome. Anyway, usually if you have the same randomization in multiple places or times, you want to re-randomize every single time if you can. And if you have an annual crop, for example, that's what you'd usually do. Yeah, I have a few other points that I'd like to still discuss. This is something that is often neglected in the experimental designs that we do in ARS, which I think can be detrimental, like the treatment by block interaction. All we've been talking about so far is accounting for differences in the mean of whatever the response is between blocks. Like this patch of land over here has higher fertility, so all the treatments are going to have a little bit higher yield in that little patch of land. That's the block effect we're accounting for. But what if the relative difference between the treatments differs by environment or by block? That because we don't have replication of treatments within blocks, the stuff I've discussed, the designs I've discussed are not very good for estimating that treatment by block interaction, which can be substantial in some cases. Like if let's say we're like looking at phosphorus addition, but like this patch of land has no nitrogen whatsoever, adding phosphorus is not going to improve yield because it's limited by nitrogen, whereas in another spot it does. So the relative difference between the treatments is different between the blocks. That can happen. And if it's possible, you can estimate that interaction better if you replicate one or more of your treatments within blocks. So a randomized block design that has more than one rep of one or more of the treatments within each block can give you more power to estimate that interaction. If you think it may be substantial, that is something to consider doing in your experimental design. Let's talk about some common mistakes and pitfalls in experimental design. Heterogeneity inside blocks, pseudo-replication, and failure to adequately randomize within blocks. Yeah, it's called block for a reason. It's not called little skinny string. It's called block because blocks are supposed to be spatially compact, because units within the block should be spatially closer to each other than they are to the ones in different blocks, because they share. That would ensure that they are environmentally as similar as possible, because things that are closer together should be more similar. So like if I see a lot of studies like this where like long rows are set up as blocks, that's not as good as a design where they're actually block shaped, as close to like a square as possible. Pseudo replication is a term you may have seen. This is a study that I had to consult on recently, where there were looking at whether free range versus cage hens had different egg quality or something, but they measured a bunch of hens or different groups of hens, but all the free range hens are in one facility and all the cage hens were in another facility. So there's really only one, the treatment is free range versus cage, but it was only randomly assigned to one entire group of hens got free range and one got caged. There's only one replication for the experimental unit. They had a lot of observational units within each of these setups, but only one experimental unit per treatment. So it's not true replication. It is not appropriate to consider all those hundreds of hens within one barn as independent, because they're all in the same barn. So they may share other confounding influences that we think is due to treatment, but it's not necessarily. So there is a resulting of confusion between what is an experimental unit. It's actually the barn. The hens are only the observational unit here. And it can, as I said, lead to confounding. And that may be okay. And logistically, this may be the best you can do. If there's only so many, it's not possible to assign free range versus cage at a very fine scale, but you need to, if you have a design like this, you have to consider what population you can make inference about. I would say it's not necessarily, you don't have a lot of statistical power to tell between the effect of aging on egg quality in this case. Yeah, but you could look at variation within the barn, but now your inference is limited to the population of that barn and not to hens as a whole, essentially. Here's another one, which I sometimes see, like this is one where people say, okay, here, I have an RCVD design for you to analyze the data from. This is what it looks like. It's like, well, what's wrong with this? Well, you can see that the blocks are the horizontal rows, but every time treatment one, and so there's 4 plots, but all the ones assigned to treatment one are on the left, and and so forth. That isn't so bad if this entire thing was replicated multiple times, but if this is the entire experimental layout, that is not good, because you have, it is accounting for variation from north to south, right? Because you have some treatment ones in the south and some in the north, but the east to west variation in the X direction is always going to be confounded with treatment. Like if there's something different about the east side than the west side, that's going to be confounded with difference between treatment 1 and treatment 4. So you have to mix up the not only the horizontal, but also the vertical positioning of the treatments. Okay, we'll talk a little about, this is just a very brief mention of statistics that can be used for designed experiments. And as I've been talking about statistical power several times, I've mentioned that we want to set up our experiment so that we have optimal maximal power with respect to some hypothesis. But what is statistical power? Well, yeah, here's a quiz. Does anyone know what movie that little clip is from? And you guess. It's from Christmas Vacation. That's right. When Chevy Chase plugs in the lights and then it, blows up the power meter there. Very good. Your corny 80s movies. So yeah, the power is the probability that you will be able to statistically detect some phenomenon that you care about if it is real. So if that effect truly exists, how likely are we with a given setup to detect that, like to get a statistically significant effect. And it depends obviously on sample size. The more replicates, the more power, and what effect it is that we're trying to estimate. But as I've hopefully shown you, it also depends on the design. And usually experiments are designed to get a desired level of statistical power. Like often it's like, okay, we want to have 80% power, meaning that if we set up this experiment many times, that to detect a certain effect of a certain size, that 80% of the time we would get a significant p-value of 0.05, usually 0.05 or less. That's what that means. And right, so that's related to the balance between false positives and false negatives. Another trade-off, right? Like if we have a stricter level of significance, or if it's a smaller, more subtle effect we want to detect, the power will go down. And what statistical models do we use for different experimental designs? Well, the basic idea is that no matter what the design is, the statistical model that we assume should represent the data generating process, meaning that anything that we think has a noticeable or meaningful effect on our outcome should be accounted for in the statistical model. Like if we think treatment has an effect, it should be in the model. If we think spatial location has an effect, it should be in the model. If time has an effect, it should be in the model. If other covariates we measure, like temperature and things, have an effect, it should be in the model. In experimental design, the structure of the units is no exception to that. And ideal tool that we have for analyzing data from these designed experiments is a mixed model. And a mixed model is called mixed because it has a mixture of fixed and random effects. What does that mean? A fixed effect, the way I think about it is it's called fixed because it's the levels of that effect are fixed from experiment to experiment. I'll explain an example of that in a second. Compared to a random effect where we are modeling the effect as being drawn at random from a statistical distribution, oftentimes a normal distribution. So I think I have an example of that. Usually treatments we model as a fixed effect, and our blocks are usually a random effect. Why is that? Like if you are testing the effect of, I'll keep using irrigation as an example, you're going to do an experiment out here on irrigation. If somebody else in another place in the world wanted to test the effect of irrigation, they could use the same levels of irrigation as you did. It would be, or if you wanted to repeat the experiment somewhere else or at another time, you would use the same levels of irrigation, or you could. Those levels are fixed from experiment to experiment. But somebody setting up the experiment in another state is not going to be able to use the same blocks as you, because blocks are one of a kind. It's like an idiosyncratic piece of land out there. They're not going to use the same blocks. You've essentially randomly drawn the blocks from a population of possible blocks you could have used, possible locations you could have set the experiment up in. So that's why we model it as random. People often ask me, should I set time as a fixed or random effect? And that depends on your question. Like if you are actually trying to look at trends over time and compare one time to another, time should be a fixed effect because you're specifically, this is not a random population of times you could have used. You're specifically concerned with a trend over time. But if you're just seeing time as a component of environmental variation that you're trying to just account for, you're never going to be able, like if you did the experiment in 2024, you don't have a time turner and can't go back in time and ever exactly reproduce the exact environmental conditions that existed at that specific time and place in the world, even if you wanted to. So it's a one of a kind random effect there, right? But I think people, while I think it's very important to set up a statistical model correctly, and that's like my job, the most important thing besides fixed versus random is just making sure that anything you think affects the response is in your statistical model somehow. That's the most important thing. Whether it's fixed or random is just sort of like a little technical difference in how it's estimated and is not as important. Although it's still something you should care about. And I'm always here to consult with you if you have a question about that. So yeah, that's basically the material I have. I just wanted to these slides are on the website, my stats website, and Axon as well. So you can you don't have to like write this stuff down, but there's some different R packages and SAS procedures that you might find useful. And R also has these things called task views, which is like a curated list of packages that follow under a certain topic. And they have one for experimental design, which is really useful to look at. Yeah, so I just wanted to give my final hot takes on everything and to put it all together. And this kind of goes against some of the stuff I've been saying. So like I've been talking about how great experiments are, but experiments are, while they are a very useful tool, are not everything. And here's a good example. So we're going back to our R.A. Fisher was kind of the hero for part of this talk, but now he's the villain because he, right. I've talked about how experiments are very important to establish causality. Observational data is correlational. You cannot necessarily say there's a mechanistic link between two things just from observational data. But that idea can be taken, that is not a dogma, and it can be taken too far. So R.A. Fisher, later in his life, kind of became a shill for the tobacco industry. He enjoyed a pipe or two himself, but he was sort of paid by them to promote the idea that that we don't have enough evidence to link tobacco use to cancer. So that was from the National Enquirer, obviously a very trusted source in the 60s. He put his views in there. And so there's sort of like a pro-experimental bias, I guess. Their experiments are put on a pedestal as like the only way to get good information. And he kind of exploited that, or I would say weaponized that idea to discredit people that were trying to establish this link. And yes, randomized trials are one of the best, if not the best ways to get information about causality, but they're not the only way to get that knowledge. There are statistical methods that if you have a plausible model of how causality works in the world, that you can infer causality from purely observational data. There's sort of a lot of techniques out there to do that. I won't go into that, but it just means that experiments are just one way in our arsenal, not the only way. And not to mention, I've said this before, but this is something you should always keep in mind. Like the biggest Achilles heel of experiments is the artificiality of the controlled conditions they impose. Even if you prove beyond any reasonable doubt that that X influences Y in that controlled lab environment, that is not evidence that it is that way in the real world. Well, it's not proof. It can be evidence, but you still have to show somehow that carries over from the lab, not necessarily the lab, but the controlled conditions of the experiment to the real world, right? Because science is trying to learn about the world. If our models don't have a correspondence to the real world in reality, they're not useful. Experiments, just like anything else. So my final recommendations for you for effective experimental design is when you're designing your experiment, explicitly think about those different components of the structure, treatment, design, and response with reference to what question you're trying to answer. Because yeah, if you want to design your experiment well, it can sort of force you to be explicit about that, and which helps your science to know that ahead of time. Keep those trade-offs in mind between realism and control, and think about where you want to fall along that, based on how many interactions you want to put in and things like that. But yeah, so put in interactions to be realistic, but don't overdo it with how many factors you have interacting, unless you're prepared for the headache of trying to interpret third, fourth order and more interactions. think outside the box when it comes to block design. RCBD is not always the best. You can get more bang for your buck with the same amount of sample size sometimes. It's not always possible to do power analysis, but I think it can be a very useful exercise to go through to actually try to estimate how much power particular designs have before you do the experiment. And this is always experiment or not experiment. You should have expectations of how the data you're going to get will look, and at least a rough idea of how you're going to analyze it, what statistical model you'll use. And having the experimental design can sort of lead you to what your statistical model is going to be. And always consult your local friendly statistician for help if you need it. Yeah, so thank you so much for your attention. And I've also thrown in some links for books that are about this topic and some online resources. Yeah, so any questions on anything? Thank you. Thank you. Thank you. Comments, questions?
So for the incomplete block design, to understand it right, so all the genotypes you are testing have to be in all the blocks at different positions.
Well, not every genotype has to be in every block, but they have to be equally likely. Well, to be a balanced design, they have to be equally likely to be in a block together with any other genotype. So each pair of genotypes has to be in blocks an equal amount of times, or as close as possible to be a balanced and complete design. Yeah, so it just enables you to get more blocks into the same physical space so that you can have finer, the more local control, because your local environment areas are smaller, so they're more likely to be homogeneous. That's why you would do that instead of an RCBD. Yes.
Yeah, for the treatment and block interaction.
Yes.
If you found their interactions, what can you do with it? Because like most of the time the block effects are random.
Yes. So you can include what's called random slope terms in your statistical model, which is essentially allowing treatment effects to be different between blocks. It uses up degrees of freedom, and you sort of have less statistical power if there's if you include that in the model, but sometimes you have to if it's needed to get a good estimate of the treatment effect. Right, like I said, so you don't necessarily have to have replication of treatments within blocks to use a random slope. You can still estimate treatment block interaction from an RCBD that only has one rep per block, but you have more power to find that interaction if treatments are replicated within blocks. But that's kind of the opposite. Like incomplete block design has less than one rep per block, but if you have only a few different treatments you're comparing, then you can actually have more than one rep per block. Like if you had a 2x2 design, that's 4 combinations. You could have blocks with 8 plots each. If that makes sense, then that would give you more power to find that interaction if it's there.
Or for, let's say, for field trials.
Yeah.
If we just measure like the plot yield,
yep.
We only got one value. But if
right.
we measure, let's say, 10 times, we randomly select 10, or we select 5 and another 5, then you have.
Yeah, so those are multiple observational units per experimental unit. Those are multiple. The plot is the experimental unit that the treatment was assigned to. So having multiple plants measured for whatever variable within a plot is not increasing the replication of the experiment. It does not have more statistical power. So if the blocking is correctly accounted, I mean, it does increase the precision. Like it's obviously better. The more plants you measure for height within the plot, the more precise estimate you have of the average height of the plants in that plot, which can increase your power overall because you have you know, they're more precise, so there's less variation between plots basically, but it doesn't increase the replication. Like, it doesn't give you more power to detect the treatment by block interaction, because you still have only one plot from each treatment combination per block, if that makes sense.
That starts people want to accept all those 5 observations, or we have to provide.
Yes, so you would include all the observations, and then using then in that case, if you had multiple, like let's say it's a block design with multiple plots per block and multiple plants per plot. First, there's a random effect for block because there's a correlation of plots within a block, but you also are going to have a random effect for plot because you have multiple plants within a plot. So there's like 2 levels of clustering basically in the data that you would have to account for. But yeah, all the individual observations. Some people like average them and then put it in the model, which gives a similar result, but it's usually better to let the model account for all of that by using different levels of random effect. Good question. Well, thank you all very much for your attention, and I'll say goodbye to the folks on Teams, and I'll stop the recording. Thank you.